Okay, so to give a brief background into this, this was an idea that kind of was fostered by a few questions from kids. And it was originally come out of a proposal to a museum for an education module that they wanted. Ended up going with something different, but then as I started to talk with people who were viewing what was done, they started to ask other questions about the human genome, which kind of led to me thinking about some of the ideas that led to I'm going to present today. So from the first presentation of this is really this idea of presenting a whole genome survey of SNPs or mutations that might occur, which may or may not be bad, which may occur at different rates, to do something in terms of trying to visualize that, to get some insight into it, to better understand what these mutations might do, where might they be located. Might they be located in highly conserved or unconserved regions? Are they connected or correlated with certain tracts? To have a visual way to explore a lot of that. And so before today, or before this weekend, no code was ever done about this, no data was ever downloaded. It was purely just an imagination I had about trying to give people a better understanding of how many variances there are in the genome in terms of mutation and how mutation affects kind of human health in general. And so with that, we decided, we thought we were going to kind of start off the project using the 1,000 Genomes project for a data source to do some analysis. And the end kind of goal of this project would be a big poster that would wrap around a room so people could, visually go through the entire genome and not only comprehend its scale, what does 3.2 billion bases look like, but then what is the variability in terms of mutations that might occur across that entire 3.2 billion bases. So that's what led to PERIL. So this was a project that we did completely with no code written at all prior to this. I don't know, should we cover some of the data aspects of it?
Yeah, sure. Okay, so when Max had originally proposed this project, I basically thought it was, with our current state of knowledge about genomic variation, that visualization across the entire genome is basically impossible, right? We don't necessarily know what every single mutation will do. They've not been nearly cataloged across the whole genome. And so there are a few resources out, and people have been working on kind of predicting the effects or even cataloging the effects of variants, but they're usually, if you want to go over the catalog, but they're usually like relatively disparate analyses, right? So if we're looking at conservation, it's only protein coding regions. Looking at changes in the coding sequence, it's only coding regions. If you're looking at 1,000 genomes data, you're not going to get any, actually super deleterious variants because all of those people are alive, and you're never going to see anything that's actually that risky. So there's really only one tool that has, in one group, that has tried to look at the effects of mutations across the entire genome, and they did this by, they called it CAD. This was done at the University of Washington. They basically used every variant effect predictor that they could find, especially for SNPs, and built a logistic regression model to predict the deleteriousness of each SNP based on all of these different databases and annotations. They then calculated the deleteriousness and scored the deleteriousness for each mutation, for each base across the entire genome. So, it's a little under, the human genome is 3.2 billion bases, but not all of those bases are actually have interesting mutations. So they broke down to, I think it's about 8.6 billion mutations. So just about all 3 possible mutations for each base across the HG19 reference sequence. And this is, really the only resource that I think could be used to accomplish Max's goal. If you're looking, at conservation, you're just going to have only protein coding sequences. Most of this plot in this visualization is going to be empty. You're only going to get some 1%. But we do want to see, some transcription factor binding site effects, which CAD included, non-coding regions, small RNAs, and whatnot. And they kind of did all of this so we get some score across the entire genome.
It basically came down to, we evaluated a lot of different ways that might theoretically show to a non-scientist what does mutation throughout the genome look like. And from everything that we had done and kind of searched through, the amount of information that goes into the CAD algorithm was the best summary kind of statistic of what would be able to consider a single base's capacity to cause some amount of harm to the individual.
And then we ran into kind of a short issue, which is that this database and this file that they provide with all these calculations is gigantic. It's about 80 gigs is their smallest version. And so a lot of the work involved kind of parsing this file and kind of minimizing the amount of relative characters that are contained in the text files that we use to generate the visualization.
So unlike most bioinformaticians, web developers want to keep their files as small as possible. We don't want big files at all. They're useless. You can't play them, you can't download them fast. And so a big challenge of this project was, how would you theoretically go about getting the entirety of the human genome over a cell phone connection or over some device that just basically downloaded off a website, right? Should we cut into demo? So with the end goal in mind being that we wanted to make a poster of this, the way that you would actually print a poster is using an SVG graphic. It's what professional illustrators use, primarily because of its SVG doesn't inherently contain any pixels, and so if I'm able to export an SVG graphic, I'm able to blow that up onto a billboard, onto any size or any printer requirements that would be available. And so rather than try to print this out this weekend or do any of that, the first step was to take a lot of the CAD metrics that we had looked at and aggregated to have some amount of an experience that would allow me to then essentially export that for further down the line. So everything we did is now available on this website that we hosted. Really meant not towards scientists as much as people who wanted to really explore what the genome is. And so right off the bat, we don't have any visualizations on here going back to the amount of data that it takes to continually download this. But you can see we have all of the chromosomes listed on the side here, so you could jump down to a specific one and kind of take it from there. So to start off with, we can click and load up chromosome 1. And so this is basically the first snapshot given the CAD files that we generated to look at this. And what's important about this is we were able to download that in like 2 seconds to get rendered out on here. And so you can look at this. Should we describe the variants that CAD breaks down?
Right. So in these visualizations, it's kind of obvious, but there are, so each base has a possible of three mutations, right? There are 4 bases in the human genome, and so CAD provides 3 scores, 1 for each mutation, and so there are differences in transitions and transversions in terms of their effects on codon changes?
So we provide a summary overview of those, the combination of those three types on the bottom here, so you can get a high-level overview of it with the three individuals broken down. And so this is chromosome 1, and unfortunately we don't have all 249 million bases of chromosome 1 on here, but we also allow pagination, so we're able to basically shift along. And so in terms of the poster presentation, I'm able to iterate over this to just continually build out an SVG graphic of the entire genome. And then also, being the website itself, we want people to be able to click through all of these other chromosomes and really kind of look at essentially, how does this abstract value of like a CAD score differ across them right online. And so, with all of these, you can highlight, go into a region of interest. And so we might pick out this one area. And looking at this is very abstract. It doesn't have very much information. But so what we do is, in addition to the 3 variant CAD scores in the sum, I also monitor and take great care in tracking the actual loci, or the position coordinates that we're at. So we can double click this at any time and go directly into the genetic or genomic position and protein sequencing services that the PDB provides, so that we can actually look at some of the gene views of this. And so you can do that with any base on here. So we can paginate through, double click on one that might be of interest, and then ask kind of more questions about it, right? Is there anything else? Oh, and finally, at the very bottom, we allow a download. If I click download, it will take quite a while because there's a lot of graphics that need to get rendered out. But so the service allows us to download all of these so that I can then print them out.